All right. Thank you, everyone, for attending the Needham Healthcare Conference. I'd like to introduce Ron Renaud, CEO of Translate Bio.
Thanks, Michael. Also, thanks for the opportunity to spend a little bit of time today talking to you about Translate Bio. Before we begin, just want to let folks know, I think this is also being webcast, on the webcast as well, that we're going to be making some forward-looking statements, and details around those forward-looking statements can be found in our SEC filings. So, a little bit about the background of Translate Bio. We may be a relatively new publicly traded company, but this is not a company that is new by any stretch, nor is the platform new. We've been a leader in messenger RNA therapeutics with more than a decade of experience focused on this messenger RNA platform. This was incubated inside of Shire Pharmaceuticals back in 2008 by our Chief Technology Officer, Mike Hartline. And since 2008, we've been really doing nothing except for focusing on how to build that platform, making it better, and increasing the focus of that platform onto diseases where we believe messenger RNA can make a big difference. We've been able to establish in vivo proof of concept studies. We've been able to have successful studies in a number of targets. We've been able to do this and generate safety as well as multiple dosing or repeat dosing. Another important feature of the platform is that right at the outset we're focused on manufacturing, and being able to make this at an economical scale is key. We've passed 100-gram capabilities on our way to kilo scale manufacturing capabilities. And this has really all culminated more recently with us bringing the first messenger RNA therapeutic to treat cystic fibrosis into the clinic, and we hope we're one of the first messenger RNA therapeutics to treat OTC in the clinic. And we're really, really looking forward to, in the not-too-distant future, clinical validation of the entire messenger RNA platform. So again, just to recap on that, again, moving into the clinic, first in clinic for chronic lung messenger RNA-based therapy. So I'm going to talk a little bit about delivering messenger RNA to the lung, not only in cystic fibrosis, but some other areas that we're interested in. To do this efficiently, we've got to be able to deliver this messenger RNA. And so we've got very strong delivery expertise and capabilities in-house. The two programs I'll spend the most time talking about today are in the lung and liver, but you'll see from our pipeline that we're looking at additional target tissues. Manufacturing capabilities, clearly critical if we want to have a cost-effective way to bring these to the market. We optimize our messenger RNA, so we want to improve its purity, and we really want to make these messenger RNAs look as much like what the body's producing on its own. And we've got a leadership team that's been involved with a number of key rare disease drugs that have spanned all the way from discovery to the pharmacy shelf. So really, really deep bench strength in terms of execution here in the rare disease space and on the messenger RNA platform. So let's talk a little bit about the platform. On this slide, really, this represents our overall business. You've got messenger RNA on the left side, and you've got the delivery vehicle on the right side. And really, if you look at this green coding region in the cartoon of messenger RNA, this is really the business part of what we do here. These are the instructions that are provided to the body to correct whatever is deficient. In the case of rare diseases, a lot of times these are mutations which instruct the body to produce the wrong protein. And so what we're doing here is providing the body with the right set of instructions in that coding region. We don't make any chemical modifications to this coding region, so we are focused on using unmodified messenger RNA to do this. We do make some non-chemical modifications, if you will, in the untranslated regions, in the capping, in the poly A tail. We also codon optimize in order to enable more efficient translation of the messenger RNA. And really, this also allows us to enhance stability for optimal expression. So once we get that part right, then we've got to package it. We've got to get it in a delivery vehicle so that it can be sent to wherever that target tissue is. The programs we'll talk about today, we want to target the lung. we want to target the liver. And so you can't just pick any LNP to do that. You've got to have LNPs that have the right physicochemical properties to be able to do that. And so an LNP that we might use in the liver or that we might use in the lung or that we might use in our vaccine program are very unlikely to be the same lipids. And in our cases, they're absolutely not the same lipids. So if you think about on this slide right here, this delivery vehicle, this ball-looking structure, which represents a lipid nanoparticle, that's what carries this messenger RNA to where it needs to do its work. And you can see that here in this cartoon. That ball, that lipid structure, carrying the messenger RNA, carrying the corrected message, goes to the cell membrane where it's captured by an endosome. That endosome takes that LNP in, and unlike gene therapy where you've got to go all the way to the nucleus and let the gene therapy incorporate into the genetic structure, we just need to get into the cytoplasm. We just need to get into where the ribosomal machinery can come in, read that message. You see those strands right there. That is the corrected message. And given that we have made a very stable and straightforward messenger RNA, one that looks as much like the body presents or produces on its own, the ribosomal machinery takes over and produces that desired protein. It can be an intracellular protein. In the case of CFTR, it's a transmembrane protein. In the cases of things like erythropoietin, that would be a secreted protein. But basically, if we put the right set of instructions in there, we can make whatever protein we want. And so you see here on slide 8, we're focused initially on the lung and in the liver. We've got a first-in-class, first-in-clinic pulmonary delivery of messenger RNA for the treatment of cystic fibrosis. And then I'm going to talk a little bit about our liver delivery program in our OTC deficiency program. OTC stands for ornithine transcarbamylase deficiency. It's a program that we filed an IND on late in 2018. We found out earlier this year that the FDA would like us to take a closer look at slow clearance time with the lipid on that. So we currently have a hold on that program, and we're looking at that, and I'll spend a little bit more time giving you the details around that. But it's a program that we're very, very excited about and looking forward to having in the clinic by the end of this year. So a little bit on our pipeline. No surprise here. I just talked about the lung and the liver. But we've got earlier programs looking at delivery to the eye, looking at delivery to the CNS. As you know, there are a lot of rare diseases in those spaces where we think we can leverage messenger RNA. And then at the end, I'll talk a little bit about our focus in the lymphatic system in infectious disease. We've got a significant collaboration, a 5 infectious disease target deal with Sanofi Pasteur. That got underway last year. Significant upfront payment. We had a $45 million upfront payment, overall $805 million deal around that. And then as a result of that, we've also begun to have a number of discussions with folks around using the same type of approach in the immuno-oncology setting. So let's dive into the CF program. I think this crowd knows the CF marketplace pretty well and knows the disease. It's one of the most common fatal inherited genetic diseases in the United States. There's a little more than 70,000 patients worldwide with this disease, just under 1,000 new cases diagnosed annually. And these patients have a severely compromised quality of life. And this is basically a disease where mucus builds up around the lungs because there is a defective protein that does not allow for chloride ions to flow back and forth across the cell membrane, and this mucus builds up on the surface of the lung. Germs collect in that mucus and leads to the deleterious effects of cystic fibrosis. And really, the treatments, there have been a lot of headway made in treatments for this disease, but they are mutation specific, and we believe there's a lot more that can be done. But there's currently no cure for this disease at this point. And so what are we focused on? We're focused on providing, again, the body with the right set of instructions to make the corrected CFTR protein. And if we're right, we can deliver that that right message to get to the lung and produce the right CFTR protein and hopefully get a more functional chloride ion channel flow back and forth across the cell membrane. And our goal to do that is with our product called MRT 5005. This is a nebulized CFTR messenger RNA and one of those little round ball cartoons that I showed you in the past, a lipid nanoparticle. And we're targeting all patients with cystic fibrosis. Remember, if you think about a lot of rare diseases, the treatments for these rare diseases are very mutational specific. They focus on a specific mutation, they focus on a nonsense mutation, but something very, very specific. Here we're providing the right set of instructions, so we're not paying attention to any specific mutation. We're paying attention to the entire disease. And the way the trial is currently set up is weekly self-administered nebulization, and we're encoding fully functional CFTR. And I'll talk about what the trial looks like at the end. But you can see in this picture right here, there's a little handheld, that little white nebulizer. That's the actual device that we're using in our current study. So very convenient. So going back to the cartoon and applying it to what we're doing in cystic fibrosis, you can see here we've got the LNP that is designed specifically to go into a vibrating mesh nebulizer. So if you think about taking a lipid, we're not just injecting this or applying this systemically like we are in our liver program. We've got to have an LNP that is engineered, that is designed to go into that vibrating mesh nebulizer, be able to withstand the shear forces of that vibrating mesh nebulizer, go into the mouth, down the trachea and into the lung, and be able to do what it's supposed to do, getting across that thick, porous mucus mesh that coats the surface of the lung. And we've designed an LNP that we believe does just that. And so that LNP carries the messenger RNA, carries the code in, that is picked up by the ribosomal machinery after the endosome breaks open and releases the messenger RNA. And then you see it gets transported to all the places that this would happen if the patient had naturally produced correct CFTR protein. Moves to the Golgi complex, the Golgi apparatus, and then that's where it's transported. The functional CFTR protein channel is transported to the surface of the cell membrane. And so you can see here over on the right, we're showing you basically all of the class 1 through class 5 defects or mutations that result in cystic fibrosis. There are roughly 2,000 mutations, about 300 or 400 of them that are followed most closely that lead to cystic fibrosis. But again, we're providing the full set of instructions here, and so we're not really paying attention to whether it's a class 1 or a class 3 or a class 5 mutation or a specific mutation. We're really looking at cystic fibrosis in the aggregate. But with that said, our initial study, our initial phase 1/2 study is focused on patients for which right now there is no treatment option. So I just mentioned a number of the modulators are focused on very specific mutations. The modulator class has made a huge amount of headway, has made just an incredible amount of change, a positive change in the lives of patients with cystic fibrosis. But there's still roughly 10% of patients for which they either produce no CFTR or they have a mutation that's very, very rare and is not amenable to treatment with the modulators. Those are the patients that we're looking at in our phase 1/2 study. But over time, the strategy, make no mistake about our strategy, it's to be able to treat all patients with cystic fibrosis. So a little bit about the data that we generated before we went to the clinic that has us excited about this program. This is an in vivo study that we looked at in CF knockout rats. So we basically have these rats that are not producing any CFTR. And in these rats, they end up generating some nasal occlusions. It's really difficult to generate an animal model that looks exactly like human cystic fibrosis. So we do the best we can with some of these various animal models, and this is one that's been recently introduced by Dr. Steve Rowe down at the University of Alabama. And what we're excited about with the results of this study is that we were able to show what you want to show in these patients with cystic fibrosis is that if you have some functional activity with the CFTR protein, you should be able to detect a change in electrical current across the surface of the cell membrane. And this study is designed to look at exactly that in the nasal epithelium. And so with these occlusions and these plugs, we were able to administer an empty LNP, so this would be our lipid nanoparticle containing no message, and then our drug product messenger, MRT 5005, which carries the correct set of instructions. And you can see here a statistically significant change in potential difference when measured in the nose. And so this really gave us a sense that we are getting the drug across those nasal or those mucosal occlusions and able to measure a potential difference or a change in electrical current. So one of the other studies that we wanted to do, and this was part of our GLP tox package that we had to file with the FDA in order to move into the clinic, was to look at non-human primates. Now, these non-human primates are wild type. These are not primates that have any kind of a defect with producing CFTR. But what we wanted to look at was, could we get these monkeys to inhale this, and what kind of protein would be generated? Would the messenger RNA get to where it needs to get to? And lo and behold, we saw just that. You can see here at week 5, and this is repeat dosing. So one of the hallmarks in gene therapy is the inability to repeat dose, and even with messenger RNA, concerns around immunogenicity for repeat dosing. Here we gave non-human primates up to 5 weekly doses. You can see here in the first 2 slides on the left, in the buffer and in the empty LNP, we don't see any protein production. At week 5, in the high dose arm, we see very nice protein production. And very interestingly here, we see 28 days after the last dose, continued staining, which tells us that that protein production is very durable. And so this is really the basis of our IND filing and how we decided to move into our clinical study. And this graphic here is a very clear picture for what our ongoing phase 1/2 study looks like. This is a randomized, double-blind, placebo-controlled, and has staggered dosing, and I'll explain to you why that is, trial looking at single ascending dose groups and multiple ascending dose groups with 3 different doses. Important to point out, this is a safety and tolerability study of single and multiple escalating doses in patients that are 18 years or older with FEV1s, forced expiratory volume 1 over 1 second, greater than 50% and or less than 90% in patients with either 2 class 1 or at least or 2 CFTR mutations. And the way we move through this study is that as we complete a dosing cohort, we need to have a DSMB take a look at it. And so once we get the green light from the DSMB, we can move to the next dosing group within the single ascending group. And then once we finish a single ascending dose, we can also move over to the corresponding multiple ascending dose part of that study. And then the final part of the study is what we're going to do at the end is we're going to pick the 2 best performing groups, and then we're going to do bronchoscopies on those patients where we'll go in and we're going to look at messenger RNA levels. We'll look at protein expression levels, and then while that bronchoscopy, while they're doing the actual brushings themselves, we'll also try to get lower airway potential differences. So again, thinking about that slide that I showed you that had the nasal potential difference, we're going to try to do the same thing in the deep lung. And so we're happy to report that we think that in the single ascending dose, we'll be ready to show data from that in the second half of this year. From the multiple ascending dose, we started dosing patients in that study earlier this year, in that group earlier this year, and we'll have data from that part of the program in 2020. So now let me move over to our ornithine transcarbamylase deficiency program, OTC for short. And this is a very rare urea cycle disorder. Of the urea cycle disorders, it's actually the most common, but this is still much more rare than what we see with cystic fibrosis. This is X-linked. It's generally seen right after birth. And what it is really OTC, these urea cycle enzymes, are the enzymes responsible for clearing ammonia from the bloodstream. And they do this at various stages of the urea cycle. And in this case, ornithine transcarbamylase, when it's not produced, can lead to a buildup of ammonia in the bloodstream. And a lot of times, these children or these infants, the parents don't know, they haven't been screened for it, they don't know they have the deficiency. And what happens is the patients either become lethargic, they can often slip into comas and or death. And what's unfortunate is even for many of these children that do survive the coma, there's often some developmental deficits that are linked to the ammonia buildup and its impact on the growing brain. And so for patients that are able to get through that, they are usually given high levels of ammonia scavengers or nitrogen scavengers. These are very cumbersome. They come with side effects, very expensive, and really don't treat the underlying cause of the disease, just treat the symptoms. And so the only curative way to approach this disease, at least at this point, is with liver transplant. And so again, what are we trying to do at Translate Bio? We're trying to provide the body with the right set of instructions to make the ornithine transcarbamylase enzyme. And that's called MRT-5201. And again, our focus here is to treat all patients with OTC deficiency. If you think about the program I just talked about in the lung, that was coming out through a vibrating mesh nebulizer where the patients are inhaling it. So think of that more as a topical approach. This is systemic. This is going to be IV administration every 2 weeks, targeting this corrected instructions to the liver. And so, again, some data here that underlies what we're excited about. This is an SPF ASH mouse model, and what we've basically done, this is a hypomorphic mouse model where the mouse produces a very small amount of the enzyme. And so to compensate for that, we put in super therapeutic doses of ammonium chloride. So really try to get that ammonia level to look like a toxic situation. And you can see here that after one dose with our 5201, we get significant increases in protein by week 1, and this is after one dose, and those levels stay sustained out to week 4. And so then when we look at what happens to the ammonia level that follows after treatment with our messenger RNA, we see plasma ammonia levels stay low out to the 4-week mark. And another biomarker is urinary orotic acid as well, also stays relatively low out to week 4. So we see this protection against hyperammonemia and normalized urinary orotic acid levels for up to 4 weeks. So this has us very excited. We think this is some of the best data that we've seen in the OTC deficiency space, at least preclinically, and gave us a lot of excitement about putting our IND together. And additionally, we're able to see very nice staining in non-human primate hepatocytes after IV administration. So you want to know that the messenger RNA is getting there. We see the very nice staining there after a single dose, and then protein detection 24 hours post-administration. So in both cases, very positive in terms of what we've seen in terms of staining here. So as I mentioned, we filed this, so we pulled all of this information together, very excited about it. We filed the IND at the end of last year. Earlier this year, the FDA let us know that after a careful review, with that LNP, so think about that again, that lipid structure that's delivering the instructions, that lipid, in the case of our OTC program, has a slower clearance time than we've seen with other programs. And the lipid hangs around for a little bit longer than we've seen with other programs. And so the FDA has asked us to take a look at that. And so what we've proposed to them is some animal studies to address that. And they've agreed that these are the right things to do. And so our belief is that we should have this resolved by the 4th quarter of this year. So our goal here is to get these animal studies done, completely wrapped up, audited, and back to the FDA with a complete response letter in the 4th quarter. But you can see here we have everything all ready to go. We have in vivo POC that's been demonstrated. All the IND-enabling studies have been completed. From a CMC perspective, we've got clinical trial supply all set. The process development tech transfer all completed. So in very good shape to move this forward. We'll complete these last 2 studies and get those submitted back to the FDA by the end of the year. And recently we've also been granted in both the U.S. and the EU orphan drug designation on this program. So another program we're very excited about. So, milestones in terms of what we're looking forward to in the CF program. As I just mentioned, I think the program or the data that most folks are keying in on right now is in the single ascending dose part of our study. And as I mentioned, our guidance is that we'll disclose that data in the second half of this year. And again, happy to announce that we have initiated dosing in the multiple ascending dose. I just talked about OTC deficiency. To get that complete response back to the FDA by the end of this year is a major goal of ours. Like we've done with CF and OTC, we want to continue to identify lead candidates for additional disease targets in both the lung and in the liver. And again, build additional partnerships and collaborations just like we did with Sanofi Pasteur. That is a program that is underway. We're very, very excited about our collaboration with Sanofi Pasteur. Off to a great start, good team, a very good rapport between both organizations on that front. Very exciting. We finished 2018 with $144 million in cash. We believe that cash is sufficient to take us into the second quarter of 2020. And we're relatively small compared to other messenger RNA companies. We're about 80 plus employees. I think our focus is going to be to get it right in the lung, to get it right in the liver, try to do it with as few people as possible, keep the burn rate very, very manageable, and as we have success in those programs, we'll expand out into other disease areas in the lung, liver, and even beyond that. And with that, I'm happy to take any questions you might have. Pardon, say that again? Yeah, so the question is the percentage of CF patients that are caused by mutations in CFTR. It's 100%. The disease is caused by mutations in CFTR. And so what you have is you have various types of mutations. In some cases, they can produce very small levels of functional protein, and depending on what the mutations are, in some cases maybe slightly higher levels of CFTR protein. And those are generally patients that are well treated with the modulators. You have a number of mutations that lead to no protein being produced at all. And those are patients where the modulators, they're just not amenable to any type of therapy. And for many of those patients, they're managed in terms of antibiotic therapy. They're managed with physical therapy to try to clear the lungs of the mucus, hypertonic saline, things of that nature. And so, these are the exact patients, at least right out of the gate, that we believe are the most important patients to try to address straight away. But the disease itself, anybody with cystic fibrosis, generally it's because they have a mutation somewhere along in that CFTR gene. Any other questions?
Hi, thank you so much for presenting. I have questions about your animal model, the clinical model. I've worked with nanoparticles before and comparing anything on the cells or RNA. I just want to know, like, do you guys have any models or evidence showing that it actually went through all the mucus and actually went into the dark express? And it seems like to me that the preclinical data would show like two separate animal models, and they're showing step by step, but it didn't show like directly like it went through the mucus, went through the cell. Can you like help us clarify? And the second follow-up question would be in the clinical studies that say that this RNA drug become one of the first RNA in the treatment for CF. Do you foresee that as a combo treatment with modulators or what?
Yeah, so two questions. The first question is really how do we know that this lipid nanoparticle containing CFTR messenger RNA gets across the mucus layer? I think that's what you're asking, correct? Based on the data that we have. So that slide that I showed you that has the CFTR knockout rat, that's about the best we have in our possession in terms of being able to show that we can get across those mucosal occlusions that are in the nasal airway and in some cases in the trachea. Those lungs are also, well, first of all, the rats are about 8 to 9 months old. Those rats also develop a bit of fibrosis, some scarring tissue on the lung. So it's not a perfect example of a CF mucus layer around the lung. but it's the best data that we have. And even if you think about the wild type mice, rats, and non-human primates that we've worked with, it's not a CF mucus layer, but there is a thin mucus layer that coats those lungs. And so I think that's the first place where we have the confidence that we're getting there. We know that in those cases, the LNP is getting to the deep lung, it's getting to the lateral parts of the lung. It's not just getting to the upper part of the lung where the lower airway meets the lung. It's getting to broadly significant parts of the lung. A couple of other things about that gives us confidence is there has been some gene therapy work using a lipid nanoparticle, delivering a vector to the patients with cystic fibrosis, a group that was over in the UK, and they were able to demonstrate that they were able to deliver this vector across that mucus layer. So we know it can be done, and the lipid that we're using, it's not the same lipid that they've used, but from a physicochemical property perspective, we believe it looks similar. So I think that gives us the confidence that we're going to be able to get there, and some of that preclinical data has borne out on that. In terms of the second question that you had is, would we use this as a combination therapy? I suppose it's feasible. I think we have to see what kind of data that we generate here. So remember, in those patients, those 10% of patients that are currently not amenable to modulators, that would be a standalone therapy. So right there, at the very least in those 10% of patients, that's a standalone therapy. Depending on what we see over time in terms of FEV1s, that will remain to be seen whether or not we need to have a combination approach. But what we do know is that for example, when we do an Using chamber assay, which is basically an in vitro assay, looking at changes in electrical current, we know that when we open up the current with a forskolin, and then in cells that are treated with our messenger RNA, and we add a modulator, we can see increases in current when we add that on top of what we're doing with our own messenger RNA. So there's a good working hypothesis that they could work well together. Whether that's necessary or not remains to be seen. Yeah, so since the introduction of modulators, these are probably questions that are much better for our friends at Vertex. But yeah, I think, they have seen improvement in symptoms. They've seen, I think in some cases the data shows reductions in pulmonary exacerbations. I think in some cases patients have shown weight gain. That's more of a systemic approach. So with the modulators, they see, for example, I think there's some data around impacts on GI absorption and things of that nature. So my sense is they've seen some improvements there. It's still really early for us to be to get to those kind of data sets. Another question in the back. Yeah, so the question is whether or not we actually use this. So you're thinking, it's almost like an ex vivo type application in lungs that are about to be you're asking about lungs that are about to be transplanted. We've been asked about that. It's not something I think we want to make sure we get to those patients right now in the data set where they've got lung functions that are above 50% and lower than 90%. Establish that there's activity there, because if you think about those patients that are ultimately eligible for transplant, you're starting to get down into that 30% FEV1 territory. And so I think what we would want to do is establish safety and efficacy in the other patient sets before we start moving backwards towards the transplant setting. But it's a good question, and it's one where because there's some very, very interesting work that's going on right now looking at CF and how to treat these transplant lungs before they're transplanted. So, another question here in the front.
Yes, I wanted to talk about preclinical work, the possibility
Yeah, so I know I have about 10 seconds here. So asking about early data that we have in the eye, the CNS, and the lymphatic system, I would tell you that pipeline chart, it's very early data as compared to our lung and liver programs. But what we've been able to do, we've got some very interesting, intriguing early data, being able to deliver some test proteins to certain areas in the eye that we want to target. Same with the CNS. And so the CNS is tricky. As you think about messenger RNA and it's the transient nature of protein expression. So those are areas that we're very interested in, and we want to be able to leverage messenger RNA for the right targets. The lymphatics, that's really where we're thinking about our vaccine approach. And so that's very active with Sanofi Pasteur on the vaccine side, very, very active. And then, I would expect that over the next 12 to 18 months, we'll continue to expand out there, whether or not that's working with a collaborator in the IO space, or we've got some interest in additional vaccine work with folks other than Sanofi Pasteur. You bet. Thank you for your time.